Yo, welcome back to the reading of Nucleogenesis. So, the Nucleogenesis Project, if you don't know, is a project that aims to develop a digital file that will represent a nucleus, so a nuclear blueprint manifest. And this project is hosted by the old Syndicate Laboratories, and my name is Dr. James Utley, your resident codebreaker data scraper. We are on part two, so let's get to reading. All right, here. If you go on the website, you can read. You can read for yourself. But here we are on part two. If you want to watch the other parts, meaning the primer and part one, go ahead. All right, so here we are. This second reading is we're going to talk about the dual stream architecture. So at the moment, biological collection, a single peripheral blood draw is deliberately partitioned into two physical and temporally distinct fractions. So if you can see that this project is based on having a dual archival and reconstruction mechanism. And so that is really outlined here in the graphic, which means that you collect a sample. One goes for all the sample processing and aliquoting and the immediate steps that are going to be needed for assays such as high C, ATAC seq, the cut and tag seq. Just because these have to be done right away because they don't measure intrinsic molecular features. They measure relational properties that depend on an intact nucleus. So that's why you got to do it right away. And then we take the other part of the sample and we inoculate or impregnate a dried blood spot pathway, and that's for the archival and future analysis. So when the technology then presents itself, because this is definitely a collect now, decrypt later type of thing, and right. So here we have kind of a nice easy breakdown. So you have your blood collection time here, and then you have your fresh fraction, which is an immediate architecture dependent analysis. And then you have your DBS fraction, which is a long-term chemically stable or biophysically stable mechanism, which then can be used in the future as well as preserved and very robust, can withstand, war, calamity, whatever you need, and whatever happens. All right. So, anyways, the fresh fraction is processed immediately to capture high entropy single structure nuclear information that is irreversibly lost upon cell lysis and desiccate or dehydration. So that's why you do it right away. These assays require an intact nucleus or a preserved three-dimensional chromatin organization at the time of collection. And that is those tests that I just mentioned. So the high C, that's a genome-wide chromatin conformation capturing to quantify pairwise contact frequencies and a higher order nuclear folding. The other one is the ATAC-seq, providing the transposase susceptible chromatin to infer nucleosome positioning and regulatory element accessibility, right? And then you have cut and tag, right, which is the target mapping of the histone modifications, preserving genomic context and positional specificity. So these are all things that have to be done right away. We're going to head over, just read to the next page, right, because I knew there was still the single cell RNA-seq. scRNA-seq, single cell transcriptomic profile to resolve cellular heterogeneity and enable downstream cell type deconvolution. So, in parallel, the dried blood spot fraction is deposited onto a cellulose based matrix, and this is the DBS part, and archived for all the low entropy, chemically persistent records of the nuclear genome. So, we just went over the ones that have high entropy that'll be lost between the lysing of cells and the dehydration. And now we do in parallel the dried blood spot impregnation or the DBS fraction, which will then preserve over time, and that is used as a genomic backup, right? So it's an independent, durable verification of the primary DNA sequence. Do you see like that's how it's used. The methylome reference, right? So that's a stable methylation profile that serves as persistent. That's a persistent epigenetic baseline by using the methylome reference. And then there's kind of a escape valve for future reanalysis. So to wrap this up together, the dual acquisition strategy ensures that fragile, high entropy nuclear architecture is captured at the time of collection, while robust, low entropy genomic and epigenomic information is preserved indefinitely. See, that's the dual architecture, right? Nuclear reconstruction within the nucleogenesis framework proceeds by integrating these temporally separated data streams. And by doing so, you're treating your fresh cell memory measurements as a structural constraint and the archived DBS data as an immutable information anchor. So that's a very important statement. So the nuclear reconstruction within nucleogenesis framework proceeded by integrating these temporally separated data streams. Now, this is done by treating fresh cell measurements as structurally constrained, because you got to test them right away, and the DBS data as immutable information anchors, right? And that's where we're going to stop today. So tomorrow we're at 1.4, and it'll be part 3. So thanks for joining me. My name is Dr. James Utley. I'm your resident codebreaker day scraper, and I'll see you in the future. Peace.